Hello, everyone, and welcome to the Thomas Expert Series. Thank you for joining us today. We're going to get started in just a moment. In the meantime, I would like everyone to double check and make sure their microphone is muted. This Expert Series is recorded and will be placed on demand within 24 hours. So please let your colleagues know how much you enjoyed the presentation today and direct them to the On Demand tab or sign up for future talks. To get everyone a little familiar with the chat, want to see where everyone's tuning in from today. I'm in New Jersey. Where are you at, David?
We're here in Kansas City, Missouri, where LabConco is based.
Awesome. We've
Philly,
got Philly,
North Carolina,
Atlanta,
Colorado, Swedesboro.
Kansas City, Florida, Houston. Got a nice mix in here today. Baltimore. All right, awesome. Everyone's awake. So at this time, I'll go ahead and turn it over to David.
I wanted to thank everybody for joining. Today, I'm going to spend some time talking about the world of biosafety cabinets and their role in cell and gene therapy, really from the laboratory side all the way through R&D and production. And today we're going to be looking at it from a standpoint, again, of biosafety cabinets. And in my role at LabConco, this is something we get involved with regularly as our products are changing and how they're used. Traditionally, biosafety cabinets have been a critical component of cell culture, and as cell culture becomes a part of therapeutics, it's a natural transition for them with some extra considerations. So just to get going here, I want to talk a little bit about the background about biopharma. And to define this category is more and more challenging because there are so many players involved in biopharmaceutical production. And really, when we're talking about biopharma, the delineation between biopharma and perhaps you could call it traditional pharma is that these are products that are derived from some kind of biological source. So it could be a basic biological components, like insulin, or it could be something much more complicated, like a cell that's actively becoming therapeutic. And these are traditionally, this is a big step away from traditional pharma in the sense that they are coming from living, breathing components, whether it be human or non-human. And this is a fairly big leap for the pharmaceutical industry, because if you look at the origins of pharmaceuticals, at least what we think of with big pharma, you can think of most of those types of products as traditional chemically based components. So you're as basic as something like ibuprofen or acetaminophen are largely chemically synthesized drugs, and those are drugs that are typically comprised of the active pharmaceutical ingredients, which is chemical, typically mixed with some binders or excipients or other things that can comprise the rest of the drug product. And typically we think about these as small molecule drugs that are not overly complicated, really, something that an organic chemist or a medicinal chemist could sketch out or derive a way to synthesize. On the other hand, biopharmaceuticals, being derived from a biological source, it suggests that there's a lot more complexity into the way they are created and to their structure, whether it's as simple as a large molecule or something more complicated like a living cell or a virus as well, or a viral vector even. And some of the examples of biopharmaceuticals, really, as simple as vaccines, which of course have been around for some time now, hormones as well, and of course immunotherapies, cell therapies, gene therapies. And it's an ongoing conversation about what pharmaceutical companies identify themselves as. But I think it's safe to say that almost all major actively researching pharmaceutical companies are now biopharmaceutical companies. So the tradition of biopharma is not exactly a new one, and a lot of products that are biopharmaceuticals have origins from hundreds of years ago. Those include things like blood products such as clotting factors or plasma transfusions. things like vaccines and hormones, really like insulin especially. These things have been around for a long time. And the transition from these over into more modern products really happened, a handful of decades ago. And you might consider this the immunology heyday of things like monoclonal antibodies, such as Humira. Obviously, monoclonal antibodies are much more specialized type of biopharmaceutical than deriving blood plasma. And these are things that are typically engineered originally in living vectors like mice, rabbits, and others. And nowadays, oftentimes made without a in vivo vector of some sort, which is a lot more ethical and cost efficient. And really today, these drugs still have immense use and are widespread therapies for a number of different things, from autoimmune disorders to anti-cancer applications. Humira, of course, probably most people see commercials regularly for this product. On to cell and gene therapies. These are much newer products and really just hitting the market within the last handful of years. And no doubt, lots and lots of focus on this next wave of therapeutics due to their impressive ability to treat or cure many different diseases and conditions. So the first ones were approved on the market back in 2017 and really largely started with CAR T therapies, as well as a couple other smaller ones along the way, such as cord bloods and others. Within this family of cell and gene therapies, it's important to differentiate the two, although the principles for manufacturing or handling these in biosafety cabinets is fairly simple. So cell therapies largely involve a collection of a cell, modifying that cell, and then reinfusing that cell into the body to give it a supercharge to allow that cell to do something extra special or in a more robust way than it would have been capable of doing so on its own. And there are generally two types of cell therapies that involve a patient's own cells being modified and reinfused, or donor cells coming from a different patient being modified and fused into a different patient. So the difference here is largely due to the ability for the body to handle rejection of those cells and managing through that, which is a very challenging process. Gene therapy, on the other hand, takes a different approach. And in this approach, instead of modifying a cell, we're really relying on modifying a gene typically through inserting genetic material or protein or other method through the use of a viral vector. So typically taking something such as a harmless viral vector, AAVs are often used in this case, and using the mechanics of the virus to apply a gene editing or a gene modification solution to a cell or otherwise inside the body. And if you look at these principles, largely the difference between laboratory and therapeutic cell and gene therapies is very closely linked. So here are two quick pictures of biosafety cabinets in use. On the left, you see a traditional, more typical laboratory style setup where we've got a technician sitting with lots of tools for research, probably overcrowding in the BSC, really some cell culture aspiration, lots of samples for running a large number of samples, all manually. And on the right, you can see users preparing a cell therapy with very similar items. We have cell culture media, we've got cell culture flasks, and really the difference here you can see is primarily between the approach with TPE and the level of focus and cleanliness on the aseptic environment within the biosafety cabinet. And many people who are familiar with biosafety cabinets or have used them in a research setting know that sometimes the practices surrounding biosafety cabinet use are a little bit lax. And that can be everything from a user's understanding of how to use the biosafety cabinet to their cleaning protocols, and even to the methods that they conduct in the cabinet, and deeming how much of that process is acceptable to conduct within a crammed cabinet versus splitting the process into separate aseptic environments. And really, as much as they are different, the principles about the product themselves are very, very similar. And that has a large focus on the biosafety cabinet's ability to produce laminar airflow to protect the product, so the sensitive cell therapy or the sensitive cell culture work, and simultaneously to offer user protection for the operator at the cabinet. And certainly when you're working with human cell culture or potentially biohazardous viral vectors, the focus on user safety is of course very important to make sure that they can conduct their work for on and on and on and not end up with an adverse effect from the therapy that they may be working in and around to produce. Really, you can summarize the difference and the expectations for biosafety cabinets between the lab and in a GMP setting as that GMP intervention. So taking all of the cell culture or viral vector scale-up principles and applying GMP expectations to the process. And that's where really the importance of the biosafety cabinet and looking at its role and how it is used comes into play. So to dive a little bit more into the function of a Class II biosafety cabinet, you can see here, there are really two key functions that I've mentioned about how these cabinets work. The first and probably most important to the GMP side is focusing on product protection for the device. So in GMPs for aseptic production, there is all sorts of focus on keeping the product as sterile as possible to reduce contamination and without incurring any insanitary conditions into the cabinet. And that's done in a biosafety cabinet by creating an ISO 5 or better environments. And if you're coming from the EU GMP classification would consider that grade A or better air. And in this environment, we're looking at downflow air that flows through the cabinet, keeps aseptic conditions constantly, sweeps any particulates or hazards away generated by the user or the process itself, and then filters that through HEPA filtration. And at the same time, the cabinet's offering that personnel protection or user protection, which is a robust barrier of air that enters from the room straight into the front grille of the cabinet, but does not enter the cabinet itself to make sure that the environment that is aseptic inside maintains itself as an aseptic environment and does not have lesser grade air flowing into the aseptic processing area. These are commonly used for a variety of reasons. The last slide showed the similarity between a cell culture activity in a laboratory and a cGMP laboratory producing cells. That's one thing, the process is very simple and similar. The other reason, of course, is cost. And the large majority of aseptic manufacturing that happens on large scale processes is either happening in automated systems or within isolators that are aseptic isolators. And for that reason, biosafety cabinets provide a great alternative for aseptic isolators because they're typically a fraction of the price, on the 1/100th of the cost of an aseptic isolator. So the cost to employ many of these cabinets is a much easier pathway for people designing out a facility. And it also processes or aligns with the process of cell therapy and viral production because those are typically smaller batch sizes, especially for patient-specific work where you're really only handling one sample inside the cabinet. So biosafety cabinets are typically not as highly customized as an isolator and therefore can be adapted at a much lower cost. Of course, it does, beg the question about the difference between an isolator and a biosafety cabinet. You can see here on the picture of the biosafety cabinet, there is a sash that has an opening with an intervention. And that means the user is putting their hands inside the cabinet. And there is some risk that exists moving from a lesser grade environment where the operator is sitting and their hands into the work area. And it's a risk assessment that must be undertaken by the facility to determine whether or not that barrier crossing can be managed with protocols, SOPs, and other practices to protect the culture or the viral vector work that's being done inside the cabinet. Now, looking at the CGT process and the flow of the research phase through production is a critical step into evaluating your equipment needs and planning for speed of the process and research in getting to market. And as we've kind of discussed in the previous slides, biosafety cabinets are a functional tool for almost any life science laboratory, and certainly in biopharma where you're handling processes that need to remain aseptic. And not only are those processes critical to keep safe and aseptic, but oftentimes the work that's done early on is going to be effectively going to be transferred over into the GMP level of work. So cells are identified early on, or a strategy is identified early on, and samples from that process are going to be purified and brought over into the manufacturing stage. So as you identify that sample and conduct substantial work with those samples, transitioning over to another set of equipment and changing the processes from the lab side to the manufacturing side can be risky without careful attention to the process. And so as this timeline is of critical importance to a drug development cycle, and reducing that time as short as possible to get the product to studies and get it to market, maintaining some consistency of equipment and processes through the entire process of R&D to manufacturing can be very beneficial. In this case, it can be as simple as looking at the scale-up procedures. Are you using Sartorius Amber 15 equipment and a biosafety cabinet that can be transferred over to the manufacturing and scale-up side as you go into hundreds of liters. Maintaining that consistency can help avoid some hurdles by changing SOPs and things like that may slow down the process. Sometimes the R&D work you may conduct happens at a level that requires the involvement of a CDMO or an outside source, and at the same time you try to manage what you can. Sometimes it's just a necessary evil to change those processes as you go to those sponsors, or the sponsor takes the work to the CDMO. And so that should be another consideration of just continuity of the process and making sure that the entire product is protected throughout the development cycle. Looking at the workflow of handling cells, this is just a generic overview and really just showing some of the role of that biosafety cabinet process in the therapy and development. And this is really looking at it from a biosafety cabinet perspective only here. So lots of things missing here, but as you can imagine, the product needs to be derived from a patient. And then at some point in time, we're going to be isolating some target cells, whether it be from blood or somewhere else in the body or a source. And that happens typically with the use of a biosafety cabinet and typically some cell selection tools. Pictured here is a very common tool, Amber 15, which is something that can fit into a customized biosafety cabinet that we happen to offer. There's also, the processes of modifying the cells and scaling up, and that can often involve automation equipment on the lab side, so liquid handling devices and small-scale bioreactors that may require larger enclosures that still provide aseptic containment. As that scale-up goes on, depending on the product, it may be very small, just a single patient, but it also could be 2,000 liters large. And in that process, lots of other processes happen, maybe not directly with the product, but in the QCing of that product before it goes back to the patient. And so that can involve lots of other equipment and enclosures, and certainly biosafety cabinets would find their use. Everything from sterility testing of the batch to the downstream kind of the QC work on the health of the entire growth phase of the cells, and so on and so forth. And then onward, biosafety cabinets continue to play a very important role. And as that product is purified and then packaged, it may be done so in aseptic conditions, oftentimes with instrumentation like a vial filler and crimping system, which you would typically find in a biosafety cabinet. And at this point, it's really, this entire process is happening on the GMP level, right? So we're focusing on complying with the CFRs for aseptic processing, and most of this work is done in the clean room setting. as a barrier to help provide extra protection for that product. Onward, that product is going to flow back to the patients. Sometimes this involves heavy involvement from the pharmacy. So that patient may undergo chemo conditioning. That's another role of the biosafety cabinet where chemotherapy may be produced, and eventually infusion of that product back to the patient, which is happening on the hospital floor, is really the time where the biosafety cabinet leads the process at the very end. And so to look at the requirements to really make sure that this process does its due diligence in protecting the therapy and ultimately the patient from undue contamination, we really have to look at some guidance for the entire process. And in the US, that's typically dictated by CFR 210 and 211, with lots of other guidance from other sources out there. In the EU GMP Annex 1, which has just gone through a massive update, is also a great resource. And sometimes you have to consider both both standards, because you may be shipping products to both regions. So these documents help really define a lot of the principles and practices for protecting cell and gene therapies throughout the manufacturing and administration process. And really, as you look to find the balance of maintaining aseptic conditions, a key preferred tool is the Class II Biosafety cabinet. And again, sometimes isolators may be used where possible. Many times, largely, we see a lot of involvement of the Class II Biosafety cabinet in compliance with aseptic manufacturing. So part of the reasons these are a favorite for the manufacturing side of cell and gene therapy processes is because of their long-standing existence as a device for providing aseptic processing conditions. So biosafety cabinets have really been around for now almost 70 years, and their design has adapted and changed dramatically in those 70 years. The key standards that dictate the design and scope of these cabinets are from NSF ANSI 49, which is a largely US-based but predominantly North American and global standard for the design of cabinets. And then in the European Union, most cabinets would follow the design guide for EN 12469. NSF ANSI 49 is updated typically about once every 1 to 2 years. EN's standard is currently actually out of date by almost 24 years. So, but they are typically just a components of assuring the device complies with aseptic manufacturing expectations. And certainly the EU GMP Annex 1 and CFRs do not specifically call out this standard exactly. They are much more focused on the environment that the product produces. And so fortunately, these are devices that meet those expectations for ISO 5 or grade A particulate-free environments. And we'll get a bit more into that in a couple of slides here. Another key part about these cabinets is that they're designed to be highly cleanable and sterilizable. And so that's critical from a contamination standpoint to prevent contamination of a batch and prevent contamination batch to batch. They also do a great job, again, of providing personal protection from CGTs, which can have a various level of biohazard. Certainly, many of the products are biologically derived, and while some of them are handled in a way or made to be less biohazardous, there is inherent risk with many of the products that are out there. And they're right size and right priced. So the batch sizes being smaller, looking at a biosafety cabinet, these are devices typically used for one person to work and process. And typically they're also much cheaper, as I mentioned, and much faster to implement, especially for large projects. So if you can imagine purchasing a biosafety cabinet for a laboratory, typically involves 4 or 6 weeks or so from start of being ordered to the day it's installed. Whereas isolators may have a 6 to 12 month lead time once the final design is actually signed off on. So speed can be very difficult as you're building a facility and getting it online. So validating biosafety cabinets is, of course, a critical step into ensuring their compliance with GMP. So the design verification for a cabinet happens through that NSF 49 process. you should really be looking at a manufacturer that has a broad scope of cabinets listed to the standard. And that ensures that there is not only diverse experience in manufacturing cabinets that are compliant with the standard, but it also makes sure that they're really staying on up to speed with the latest developments in the standard, and to make sure that their whole product line produces, you know, a level of quality that would match the expectations for a cell therapy. As you move away from determining that cabinet, the process of actually validating it in your facility includes a couple of key steps. Starting with, at the end of manufacturing, a factory acceptance test report. And that's something that the manufacturer that's going to be complying with NSF 49 is required to do to document the cabinet's compatibility with its manufacturing relative to the standard and its suitability for the end user as a aseptic containment device. Once a cabinet gets to a site, there's the all-important process of an IQ/OQ step, and that's the installation and operational qualification to make sure that the device is installed correctly within the manufacturer's specifications. And then the PQ step is another critical step for equipment of all kinds touching GMP. And this is a process with a biosafety cabinet that is no different than an isolator, where you would have a skilled technician who's capable of testing airflows and HEPA filters for leaks, and so on and so forth. Have them come in, validate the equipment to make sure that it's doing all of the parameters that it's supposed to for aseptic conditions. And this is information that's typically documented on more modern BSCs like ourselves on the software, or through dry contact outputs with alarm conditions and other states that can be communicated after the certification is completed. So IQ/OQ, PQ is a stack of paperwork, and from there, ongoing maintenance by the cabinet itself helps make sure that the cabinet stays in check between each certification every 6 months in a GMP environment. From another standpoint, of course, you want to be working with manufacturers who have the quality controls in place to make sure that they're documenting all changes and activities happening by the organization. LabConco obviously is someone who's ISO 9001 compliant, and this is just a routine general expectation for any equipment, whether it be a door hinge to a biosafety cabinet touching a GMP facility. Onward for the biosafety cabinet to perform once it's up and running. Again, this is a device that is going to be producing sterile airflow, and as a product made in a biopharmaceutical facility is going to be a much more complicated, sensitive product. It's typically something that cannot go through a final sterilization or filtration that will rid it of contamination. And so that's why there's so much focus on protecting the environment in which the enclosure is, or the product is made, from the clean room to the aseptic site itself in the biosafety cabinet. And so when we're looking at those standards, ISO 5 is something that would comply with ISO 14644-1 and 2, and grade A again for EU GMP. That's where we would look at for definitions for aseptic conditions in terms of the particulates in the air and viable contamination in the air. This is a process that's done through environmental monitoring, which is a family term which helps describe the processes for looking at contamination in the room. And so here's a picture of someone taking an air sample in presumably the corridor or hallway of a aseptic manufacturing facility, which is just a part of the environmental process, monitoring process. So there are two real main components that occur and touch the biosafety cabinet. The first part of the environmental family is viable sampling. This is something that looks for particles or contamination that is living. So, or really a larger biological structure. So typically things like mold and bacteria. And the way those are sampled for is done with a tool that is going to actually support the growth of that contamination once it's collected. And so on the top left here, we've got a picture of a basic TSA settle plate. It's the older, kind of more classical approach for monitoring viable conditions. On the right, you see an image of active viable sampling device, this would be something that would be connected to an instrument from a typical environmental monitoring company and would pull a known CFM onto the plate that would then be incubated and evaluated downstream. So that process looks for viable contamination. Of course, viables are just one part of contamination. The rest of the contamination would be considered non-viable and may include everything from dust and dirt to, again, mold and bacteria. And the measurements for non-viable contamination are done through particle counting in that same environment. So here we see a picture of a typical particle counter. This one happens to be from TSI. And this is a device that will pull a known volume of air in and assess the amount of contamination in that air at different particle sizes. And depending on that level of contamination, that will correlate directly to the level of air quality in the environment and whether or not it complies with the GMPs for producing an aseptic product. There are a couple different ways to integrate this equipment into biosafety cabinets, and the way this is done is a very rapidly changing, how do I say, it's a rapidly changing kind of design, and ultimately the component that has to be integrated into the cabinet is that collection source for that air sample. So on non-viable monitoring, you need a funnel, which is called an isokinetic probe. It's a very specialty shape and size, depending on the airflow, velocity, and volume at which the sample is supposed to be collecting for the particle counter. And you can see here that we've got a non-permanent and a fixed installation. On the top, the non-permanent installation is basically adapting a cabinet that was not anything customized when it was ordered. So making use of a pass-through to run a big, large bulky cable into the center of the work area. And this is a device that has to reside there during the entire aseptic processing process. So you can imagine, someone who might be tight on space or does not want to clean all of the areas in and around that tubing may find themselves a little bit frustrated with this design. It also can move. And as a part of aseptic processing, you need to establish a solid place to monitor your samples. If this can be moved around, it can be difficult to ensure the consistency of that air sampling measurement point. So down on the bottom shows an option for a hard welded design. And this allows just for a lot more simplicity in biosafety cabinets that are being monitored or that are monitoring airflow for non-viables. There's an article on our website. If you search for more information, just search particle counting in a biosafety cabinet, you'll stumble upon lots more information around these options. On the viable side, really the sampling conditions are very similar. So you can either choose to have something that is loosely set into the cabinet or fixed in place. And the settle plates that I mentioned earlier is as simple as putting a TSA plate on the work surface and leaving it open. However, that same design can be fixed and integrated with a fixed location on the back wall or a place that's most convenient to the monitoring process. And again, this just helps permanently affix the environmental sampling location. And the reason that's so important for these processes, again, is because as you're establishing a protocol and an SOP, it needs to be consistent and repeatable for inspectors and the manufacturer alike to verify the quality of the product. So there are some guidances for this, and they exist in the GMPs to show where the process should really be located. And the location largely has to do with the ability for that sampling device to collect an appropriate sample within the cabinet. So each of these devices needs to be within 300 millimeters of the process. And the process is defined as the area of activity where the aseptic process is occurring. And so here you can see, we've got on the top, middle, the isokinetic probe maybe is affixed there. And maybe if you're using a TSA plate, it's set just aside from that. And really these should be located, at or slightly above the level where the aseptic process is happening. So having a settle plate sitting on the work surface really doesn't create a great sampling location because it is at a lower height than where the aseptic process is likely happening. And that's just because users are going to be working at maybe 8 or 10 inches off the work surface in the biosafety cabinet. So again, hard fixing these into the cabinet brings some dedicated location, keeps the area where the aseptic processing happens a little bit more rigid. And if you're wondering to yourself, well, there's really not much space to work if you have to work within 12 inches of the process, that can be accounted for by adding more sampling locations within the biosafety cabinet to make use of a larger area, or maybe the process is just small enough that it fits in that area as well. But that's an important step to consider as you're laying out a BSC for a GMP environment. Now, validating aseptic conditions really is a process that's a ownership by the manufacturer of the drug product. But in general, there's some comparisons that are important to note, because there are some differentiations that exist between the R&D side and the GMP side. And I've added hospital infusion pharmacy in here as well, because it lives somewhere between a research lab and a GMP facility as well. So, for the biosafety cabinet, or hood, as I'm calling it on the slide, typically biosafety cabinets are going to be certified every 12 months in a lab. And then on the sterile manufacturing side, in a pharmacy or in GMP, it's typically done every 6 months. So that ensures routine, consistent aseptic conditions and airflow, and a much shorter duration between inspection by a technician. The user process, when you're validating how somebody does their job and ensuring that it's done correctly, in a laboratory, you may have some general guidance, you may have no guidance. In a pharmacy, there's an expectation that there's skill certification and recertification. And on the GMP side, there's a heavy focus on SOPs, training, retraining, and focus on maintenance of aseptic conditions. Now, the batch, the environment that the batch is produced in, really there's no regulatory expectations for the research lab. And in a pharmacy, there's only light duty testing as well. So oftentimes the ISO conditions for the room are loosely calculated, and there will be some inspection and environmental monitoring of the pharmacy. But in a GMP setting, there's typically routine work going on. And for the batch itself, product that's manufactured in these environments, that's where you're going to see a lot of focus only on GMP for the product to be inspected and QC tested after it's released. And really before it's released, of course, as well, to ensure that it's not only suitable for the patient, but it remains aseptic on the shelf. Sometimes you see aseptic checks on pharmacy products that are sitting on the shelf much longer, but it is rarer depending on the type of work being done. Looking a little bit more in depth at cleanability, this is another critical area for GMP. And while a laboratory is typically going to select a disinfectant and cleanability approach that's based on effectiveness of eliminating a bacterial hazard or viral hazard as they work also towards contamination, the GMP expectation is going to take that to a much more in-depth level to make sure that not only is the product being removed, but there's also no contamination from other factors such as spores, mold, or other contamination that can otherwise cause grave impact to the patient receiving the therapy. And so the routine expectation is that the facility is going to be producing at a minimum in a full clean room suite. Typically, these environments are highly engineering controlled, with HEPA filtration and ISO 7 or better expectations for the room air quality. And then the expectation for users in these environments includes lots of garbing and equipment that's also really designed in and around regulatory expectations for cleaning. So lots of stainless steel, cleanable casters, really durable materials that will stand up to aggressive cleaners. And transitioning that over to biosafety cabinets, that really, means that the biosafety cabinet itself must include those same types of expectations for cleanability. And that means, matching the materials of construction with batch agents that are being used for cleaning, such as spore cleanse, Vesphine, Paradox, and other, sporicidal agents that can be really aggressive and destroy lots of materials if they're not stainless. So I mentioned here that oftentimes biosafety cabinets are made from largely from 304 stainless steel on the inside. And it's acceptable to use these devices, but they can rust with improper treatment. So it's just a routine expectation of the manufacturer of disinfectants like Spore Cleanse. And if they're going to be used, they need to be used properly with a secondary cleanup using a sterile IPA to remove aggressive residues. So LabConco does offer lots of different features in our biosafety cabinets that help accommodate regulatory expectations for cleaning. So that includes, simplicity in our design, having a one-piece dish work surface that you can clean easily, doesn't, harbor any leftover disinfectants. The inside of our cabinets also have lots of other flush, easily wipeable areas. The outlet covers, for instance, are completely flush. And the cabinet can also be routinely disinfected and sterilized using VHP systems that can be connected either to the entire room itself or directly to the biosafety cabinet. And in some cases, if the design is focused on extra reliability for cleaning, we're able to do other modifications that include stainless steel exteriors and other modifications that promote cleaning. As you're considering that space, here's an image of one of those examples of just a cleanable environment that we can create with some extra engineering components on the top of the BSC. You also need to think about not just the smooth, easily wipeable areas of the cabinet, but also ensuring that there's proper space around the cabinet, and really focusing on not just the quality of the construction, but also where you're integrating these devices and facilities. And so that's a step where planning with your architect, planning with the manufacturer of the cabinet, and really having a holistic approach to implementing a biosafety cabinet into the facility can really make for a much better successful rollout of your clean room as you're making cell and gene therapies. And so with that, I'd like to conclude the discussion on biosafety cabinets and cell and gene therapy. And I'm going to turn it over to questions at this point. And I already see at least one came in with respect to cleaning. So I'll jump in and read this one and go ahead and answer it. So Miguel asked a question about disinfecting stainless steel surfaces. And it talks about, is a wipe with IPA of the surface after a sporicide recommended? And the answer to that is, it really depends on the disinfectant. So, most manufacturers, whether it be someone like Ceres or Contec, those are great people to reach out to about chemical compatibility with your specific disinfectant. I can tell you many times, most sporicidal cleaners, if they're not followed up with a secondary wipe with a sterile IPA or some other deactivating agent, can and will rust all types of 316 and 304 stainless steel over time. So it is highly recommended to follow the manufacturer's guidance, but largely we have seen after that appropriate contact time that coming back with a secondary IPA that's sterile will drastically improve the lifetime of stainless steel in these environments. Let's see a couple other questions here. So Rhonda asks, does LabConco offer stainless steel exterior options for biosafety cabinets? The answer is yes, we do. This is predominantly for GMP manufacturing environments. They do come at a slightly longer lead time and cost, but it is something we do provide. And so really go hand in hand with the expectations for cleaning. So as you can imagine, a stainless steel surface is going to be about as robust as you can expect for sporicidal agents. Let's see, Diane asks, you have we have a capper crimper machine that's too large for a biosafety cabinet. Do you offer other solutions for these devices? It's a great question. We do, in fact, offer larger biosafety cabinets that are customized for equipment and larger processes. I don't have an image on this slide. Let's see if I can go find one. The model we most regularly provide would be the Logic View, and that's a larger biosafety cabinet that's shown up here on the top right. This is something that's about 42 inches on the interior and really designed around automation equipment, cappers, crimpers, fillers, and other affiliated devices. Oftentimes we see users request these where they would like someone working with a repeater pump on the left, filling aseptic vials, or someone working with a repeater pump on the right, filling syringes. And then we and then we'll pass through a pass through to another technician who would be capping and crimping the vials in a different device in a different environment. Let's see. Sarah asks, do we offer cabinets that fit Sartorius Amber 15 instruments? The answer is yes. Actually, I don't have a picture of one on the slide, but here's a Amber 15 24-way, and regularly we're providing biosafety cabinets for these devices because they're exposed bioreactors that have to remain in aseptic conditions to protect the cells as they grow. So we do a number of different modifications to support this type of instrument, and from our perspective, it would be one of the most common devices placed into a biosafety cabinet at the earlier stages of biopharmaceutical development. Great questions, everyone. See, Jackson asks, what's the schedule for performing an inspection of a biosafety cabinet? Is it yearly? And how much time does it take to perform an inspection of a biosafety cabinet? The answer to this question is slightly dependent on the regulatory environment you comply with. So for most laboratories and non-GMP or non-hospital pharmacy settings, the typical schedule would be done at installation, and then a certification would happen then again every year. That process involves leak scanning the HEPA filters for any problems. It involves setting the airflows of the cabinet to make sure that they're doing their job to contain and protect everything from the user to the sample. And then it involves a generic inspection of the laboratory space and smoke study, typically of the sash and containment area, to make sure that the cabinet's working correctly. That process typically takes about an hour. The fastest, you would not want to see it done any faster than 45 minutes. That would be very fast. So typically an hour, hour and a half is typical. For GMP or pharmacy settings, there's typically extra steps that take place. So, and those happen on a routine schedule of every 6 months. They would be doing environmental sampling, and that process can take sometimes an hour and a half to 2 hours per hood, just depending. Great question. Don't see any other questions at this time. I'll maybe give it another 20 seconds or so, and then I think we'll wrap it up.
Yeah, if anyone has any last minute questions, go ahead and type them in the chat. But for the moment, I do want to go ahead and share my screen with you all just for a second. Wanted to take a moment to show you that there is a giveaway if you take the survey in LabConco's kiosk. There's also literature, web links, as well as videos in here. So be sure to check all of those out. We also have our on-demand talk. So this talk will be on demand within 24 hours. So it will live in here if you guys wanted to come back and check it out again. And there's also a bunch of other talks in there. And then we'll go ahead and check out the chat and see if anyone else had any last minute questions. Not seeing anything.
I'd love to give away a biosafety cabinet. Maybe the next session.
That would be nice. All right, everyone. Thank you so much for joining us today. I hope you all have a great rest of your day.
Again, all right. Bye, everyone.